Okay. In yesterday's class, we have completed amoebiasis. Disease caused by which causative organism? Chat. Amoebiasis or amoebic dysentery is caused by which protozoan? Quick. Very good. Entamoeba histolytica. And we have started Trypanosoma gambians. And we'll finish some of its important things. Systematic position, and there are different forms of trypanosomes, polymorphic forms. That is leishmanial. You have to remember the other name of these different forms. That is amastigote and the important unique characteristic features of each forms. Okay, and just remember the picture. If you remember the picture, it is very easy for you to remember the characteristic features of every forms, right? And leptomoda means promastigote, critidial means epimastigote, trypanosome means typomastigote, like that. Okay, and we have seen where it have been who have been discovered this and where it have been distributed, and where then important things about the habit and habitat and their morphology. And they have a pellicle flagellum and attached flagellum, free flagellum, undulating motion and undulating membrane, cytoplasm, and their nucleus. Up to this, we have completed. Okay, in today's class, we are going to start from metabolism of Trypanosoma gambians. Okay, in today's class, we are going to finish the protozoan diseases. If not, by tomorrow, everything about protozoan will be completed. And after finishing the protozoan, we are going to solve and I have give you some test. Okay, live test, and you have to answer all those questions, and you have to count the marks, and you have to analyze yourself. Clear? Okay, let's continue. In today's class, we are going to start the metabolism of Trypanosoma gambians. Okay. In the absence of mouth, the trypanosome obtains its nourishment by osmotropy from the blood plasma. I have keep on telling multiple times what is osmotropy. And this osmotropy works on the principle of osmosis, that is movement of fluid or the solvent or the solute from the higher concentration to the lower concentration. Okay, that process is called osmotropy, and the organisms which follow this osmotropy is called the osmotropes. And trypanosoma also obtain its nourishment when in the absence of mouth, the process of osmotropy. In its long slender form in the human bloodstream. In bloodstream, it looks in the long slender form, and it absorbs a large quantity of blood glucose. And after absorbing this glucose, it undergoes these glucose have been converted to the pyruvate, and that process is called as the glycolysis process. We have read in the biochemistry that conversion of glucose into pyruvate in the presence of oxygen, that is called as the glycolysis process. But here in the anaerobic process, this glucose is converted into lactate. Okay, and this is also a process of called the glycolysis. And why this glycolysis has to happen means, because when it undergoes the process of glycolysis, they are tend to release some of energy, and that energy is taken for the organism or the protozoan for its metabolic activities. And at this stage, the mitochondrion is without cristae and thus inactive. See, when the mitochondria is present without the cristae, it is inactive in nature. And that is how at this stage, the mitochondrion is not having the cristae, so it is in the inactive form. Okay. Under certain conditions, when blood produces antibodies to prevent the absorption of glucose, when the cell finds that absorption of glucose is enough, then the blood produces the antibodies, and this antibody is going to prevent the absorption of glucose. Now the mitochondria is going to develop the cristae. When the mitochondria start developing the cristae, it becomes active. Once the mitochondria becomes active, they tend to release several enzymes. They undergoing the metabolic process. See, in the enzymes in Krebs cycle, we know that cytoplasm is the area where the glycolysis is going to happen, and the mitochondria is the place, especially the cristae that or the inner membrane. That is the area where the Krebs cycle is going to take place. Okay. Now, the enzymes in the Krebs cycle oxidizes this pyruvic acid. See, once under glycolysis, the glucose has been converted into the pyruvic acid. Now, that is up to the glucose is converted into the pyruvic acid. That process is called as the glycolysis. And once the pyruvic acid converted into another product, that is acetyl CoA, that is said to be as a Krebs cycle. What is going to happen in the Krebs cycle means they are going to oxidize this pyruvic acid. This is the product of glycolysis, and once the pyruvic acid has been oxidized, it releases the carbon dioxide and water and yield some of its energy. Okay, in the presence of oxygen, that is aerobic form. Now, at this stage, the trypanosome stops multiplying and takes up the first intermediate forms and then slender stumpy forms. And you have to remember what are all the different forms of trypanosomes. Okay, and what is called as the long slender form and what is called as the short stumpy form. I have discussed everything in the yesterday's class, and once this has been over, once I have taught every diseases, I have sent this, I have uploaded this PPT in the WhatsApp group, and you can revise those things. Okay. And in the body of sepsi fly, the trypanosome forms in the midgut and ruthedial forms in the salivary glands. See, I have explained in the yesterday's class, there are two different forms in the sepsi fly will be occur in the bloodstream of the vertebrate host. That is, in man, the trypanosome may be long, slender, or short, stumpy, or intermediate between the long, slender, and short, stumpy form will be occur. Where in the vertebrate host, that is man. In the invertebrate host, that is called as the tsetse fly or carrier, the trypanosome is long, slender in the midgut. When it reaches the midgut area, the trypanosoma gambians is in the trypanosome form, that is in long, slender form, where in the midgut of the tsetse fly. But in the salivary glands of the tsetse fly, it is in the short stumpy form. These are all the things you have to remember. And in the salivary glands, what is the name of the form? Critidial form. In midgut, it occurs in the trypanosome form. In the salivary glands, it is occurs in the critidial form. That is short stumpy form. Okay, you have to remember these things. Okay. In the body of tsetse fly, the trypanosome forms in the midgut and critidial forms in the salivary glands have the extensive network of cristae containing the mitochondrion. As the parasite ceases to get the blood glucose and thus depend wholly on the pyruvic acid. What we have said, when the cristae is absent in the mitochondrion, the mitochondria becomes inactive. When the mitochondria starts developing its cristae, it becomes active and it going to synthesize a series of enzymes, and that going to convert the pyruvic acid into the carbon dioxide and water, and that is going to generate many energy, and that energy is going to taken by the organisms for its further metabolic activities. Okay. Now, the metacyclic trypanosomes in the salivary glands do not feed. See, they do not feed. Which form of the trypanosomes? Metacyclic trypanosomes in the salivary glands. They do not feed, and their mitochondrion assumes a crystalless tube, possibly indicating there is a reversion to the mitochondrial activity at this stage. See, once they have completed all the above process up to this, in the metacyclic this is the next form of the trypanosomes, that is metacyclic trypanosomes that is present in the salivary glands. They do not feed. As they do not feed, they don't need any requirement of energy. As they didn't need any requirement of energy, the mitochondria has lost its cristae. And once the mitochondria lost its cristae, their mitochondria becomes inactive. They are in the inactive stage. Okay. Now, the DNA of kinetoplast plays some yet unknown role in the mitochondrial role. See, we know that mitochondria and the chloroplast is said to be as the semi-autonomous organelle. And here the DNA of mitochondria is going to make, definitely they are going to synthesize some of the proteins, but that function is not clearly known. And the gaseous exchange in the respiration and the elimination of excretory products takes place by the diffusion through the pellicle. But then what is the excretory organ present in the trypanosome means? There is no particular organism, and that is the excretory product eliminated by the process of diffusion. That too through the pellicle. Osmoregulatory mechanism is altogether is absent. Although it is a protozoan, we have said that contractile vacuole is present in the many of the protozoans, and that is responsible for doing the osmoregulatory function. But here in trypanosoma, this osmoregulatory mechanism is absent because contractile vacuole is absent here. That's all about the mechanism. In here you have to understand what are all the process they are going to follow to synthesize the energy. First one is glycolysis and the second one is Krebs cycle. Okay, this is what you have to remember. And what are all the forms which do not undergo any feeding mechanism, that is metacyclic forms. And another thing is what are all the two different forms which is present in the sepsi fly and which forms is present in which area. In case it is a trypanosome form, it is in midgut. In case it is a critidial form, it is in the salivary glands. If the mitochondria is present with cristae, it becomes active. Once it loses its cristae, mitochondria becomes inactive. These things you have to remember, none other than. Okay, now reproduction. What is the reproduction type seen in the Trypanosoma gambians means asexual reproduction. The sexual reproduction is unknown in this type of species, Trypanosome gambians. Asexual reproduction takes place that too by the binary fission. Now, how the reproduction has been going to take place? First, the kinetoplast and the blepharoblast divides to become paired. They are going to divide and they became paired. Kinetoplast and the blepharoblast. The old flagellum remains attached to one of the blepharoblast while a new one begins to grow out from the other blepharoblast. Now, the nucleus divides by mitosis process. This mitosis is the type of a cell division, and this cell division osmoregulatory is absent means, see what I have seen here. What is the function of osmoregulation? Okay, there is no, this is done through the body surface. As I have mentioned, everything has been done through the pellicle, not a particular organism. Contractile vacuole is absent here. Clear? Here, a specialized structure pellicle is present. But take in case of amoeba, it has no a definite structure. It because pellicle is absent, there the requirement of contractile vacuole is present. Okay, but here the diffusion has been takes place through a pellicle. Okay. The nuclear divides by the mitosis into two daughter nuclei. We know that mitosis is the type of cell division. Once the nucleus divides, what is the other name of nuclear division? Karyokinesis. Once the cell division starts, first initially the nucleus will be divided. Following the nucleus division or the karyokinesis, the cytokinesis will happen. After the nuclear division, cytoplasmic division will happen. That is the two daughter nuclei will be present. That is the end product of the binary fission. We know the mechanism of binary fission. Finally, the body splits mid-longitudinally from the anterior and backwards, forming the two daughter nucleus. In this slide, what you have to remember is the reproduction followed by trypanosome gametes is asexual reproduction. That too by the binary fission. Now, what are all the two important things? Plays a major role in the reproduction means kinetoplast and blepharoblast. Okay, and another one is this point. The old flagellum remains attached to one of the blepharoblast, and while a new one begins to grow out from the other blepharoblast. That is, we can say that binary fission division, and the mitosis is going to happen as usual. Mechanism will be followed. Okay, that's all about the reproduction. And coming to the life cycle, and what see when it comes to the diseases, what are all the things I'm highlighting? Just make a note, that is more than sufficient for you. In this slide, you have to remember what is the host. And trypanosoma gambians is digenetic in nature, which means what? It needs two different hosts to complete their life cycle. One is the tsetse fly. okay, as its carrier, and the primary host, that is human. It requires two different hosts to complete their life cycle. That is why it is called as the digenetic to genetic. To complete their life cycle, it needs two hosts. So this is an important thing you have to remember. First important. And the second thing is a primary or the principal host. And the primary host is also called as the principal host, and it is also called as the definitive host. What is meant by this definitive host means an animal in which the parasite feed and attain the sexual maturity. That is why I keep on stressing primary host is the place where the parasites undergoes a reproduction. Okay, where it attains its sexual maturity. That is why it is called as the primary or principal or the definitive host. Clear? And for this Trypanosoma gambians, the primary host is man. Remember, this is the second important thing you have to remember in this slide. And the intermediate host or the vector is a blood-sucking insect. Blood-sucking insect called the tsetse fly, and the scientific name of the tsetse fly is the Glossina palpalis. Okay, and you know what is the intermediate host and what is the reservoir host. And what is the reservoir? These are all the intermediate host. Means the intermediate host is the one going to act as a carrying vehicle, or we can call them as a vector. And this vector is the one going to carry the parasite from the reservoir host to the primary host. Clear? And what is that intermediate host here for the Trypanosoma gambian means tsetse fly. That is Glossina species, particularly the Glossina palpalis. And the reservoir host, that is to make the parasite temporarily active until it reach the reservoir host or until it reach the primary host. That is called as the reservoir host. In reservoir host, the parasite doesn't undergo any of the vegetative changes or as well as the reproductive changes. Neither of the changes have been happened for the parasite in the reservoir host, as well as it doesn't cause any of the metabolic changes in the reservoir host also. That is why it is called as the reservoir host. The only thing made by the parasite is to make them temporarily active until it completes its life cycle. That is reservoir host. And the mammals, particularly the mammals, act as a reservoir host. Antelopes, pigs, buffaloes, they are act as a reservoir host. This is the third important point you have to make a note of. Just make it is a digenetic, and the primary host is human. The intermediate host is the tsetse fly, and it is blood sucking insect Glossina palpalis. And the reservoir host is mammals. These are all the examples of those mammals. These are all the three important points you have to focus in that slide. Next, life cycle in man. Before that, I have to show you a picture. Okay. So just see here. In this picture, the question can be arised. And what are all the things I have mentioned here that has been written in the theoretical form? See, this is the tsetse fly. This is the area where the parasites in the tsetse fly. And this is the area where the parasites is present in the bloodstream. And what are all the things you have to focus here is in man, where they have been inhabited in the bloodstream. In the insect, in the tsetse fly, where they have been inhabited means salivary glands as well as the gut. And you have to remember what are all the things happen within the gut and what are all the things happen in the salivary glands, what are all the things happen in the bloodstream of the human. See, when we know that from the insect, they have been carried to the human. So we have start from this tsetse fly. After the bite of the tsetse fly to the reservoir host, they have been taken the parasite within the body of the tsetse fly. And after this, what is the form which is present in the midgut of the tsetse fly? Trypomastigates enter the tsetse gut. What is the form? Trypomastigate. And this trypomastigate has been converted to the procyclic mastigotes. Okay. And this procyclic trypomastigotes will be converted to the epimastigotes. And these three things will be happened in the gut of the tsetse fly. And after this epimastigotes will be converted to the metacyclic trypomastigotes in where? Salivary glands. Clear? And from this, they have been remain in the same place. Salivary glands. This insect will keep this parasite in their salivary glands itself, as in the metacyclic trypomasticates. Okay, and now this insect, this tsetse fly, will going to bite the human. After the bite, the parasite which is present in the salivary glands of the tsetse fly, which is in the metacyclic trypomastigotes, will be enter into the human in the metacyclic trypomastigote. Okay, and human, they have been enter into the bloodstream. Okay, after they have been enter into the bloodstream, they become sexual maturity. They have attained their sexual maturity, and they have been they have undergo the reproduction, that to asexual reproduction, particularly the binary fission. And after the binary fission, they have been converted to the trypomastigote form. See, here the trypomastigote forms, procyclic forms, and epimastigote forms. In the salivary glands of the cystifly, it is in the critidial forms, and that is called as the metacyclic trypomastigotes. But here in human, the trypomastigote form will be present here. Okay, this is the life cycle of the cystifly. This is more than sufficient to read this Trypanosoma gambians. Okay, that's all about the life cycle. Now come back to the theoretical part. Life cycle in man: infection is always initiated by the bite of the tsetse fly, that is Glossina palpalis, which harbors the infective metacyclic forms in the lumen of its salivary glands. Just now I have explained it. Have one always the infection will be start only by the bite of the tsetse fly. Okay, and when it bites the human, the metacyclic forms which is present in the salivary glands will be enter into the human lumen. Okay, after it enters into the human, it reaches the bloodstream. Now, when the fly, you have to remember this metacyclic form is the infective stage. This also can be asked. When the fly feeds, we know that this fly, tsetse fly, is a blood-sucking insect. When the fly feeds upon the man's blood, it releases the contained trypanosoma into the bloodstream. Okay. Now, while feeding, the fly grips the skin surface with its claws and punctures with its proboscis. And how the fly will puncture the skin of the human means with its proboscis. And the salivary glands, when it bites the individual, the it always, see, this is the mechanism. You have to remember the same thing will be applies in the malaria also. See, this is the insect. Consider this is the insect. This is going to bite the human hand. Consider this is the surface of the human hand. See, it has to bite, and the blood has to came out. We know that the blood, once the blood will make a contact with the air. See, once the blood came out of the human surface, came out of the human, it makes a contact with the air. What is going to happen? The blood will coagulate. Okay, and I will have to ask you one more question. What is the anticoagulating agent? Why the blood will not coagulate inside our body? What is the anticoagulating agent? And or just tell me the reason why the blood will not coagulate inside the body. Once it makes into contact with the air, it immediately coagulate. What is the reason? Just tell me fast. This is the basic thing. Let me repeat my question. Why the blood is not coagulate inside our body? It keeps on. It is in the it is in the fluid-like structure. It is running here and there. But once it makes into contact with the air, it becomes coagulate. Just say me yes or no. Lalita, Anjanelu, Aruna Jyoti, just answer for this question. Just say yes or no. Do you know or what? Okay. See, do you know? Do you heard about the name heparin? Do you heard about the word heparin? This is the anticoagulating agent which thrombin, they are the clotting factors, but the exact word is heparin. See, this heparin is the one makes the blood to prevent from the Coagulation. See, this heparin is an anticoagulating agent. As the heparin is outside of the body, it makes into contact with air, it becomes coagulate. So, once the insect will bite, the blood will came out. Once it came out, it starts to coagulate. So, to prevent that coagulation, this insect, the salivary glands, it has to send some of the salivary glands, it has to set some of the saliva into it. In the saliva of the insect, as well as whatever be the blood sucking insect is there, always once it starts to suck with their proboscis, the blood will come out. The blood immediately starts to coagulate. To prevent this coagulation, they have to send some of the saliva in it. And what does the saliva contain? It contains some of the anticoagulating agent like heparin in the insect of the body. It may be any of that. That an anticoagulating agent which is present in the saliva will make this blood to prevent from the coagulation. Okay, now when it sends some of the saliva, the parasite which is present in the saliva will also reaches the body of the human, and from the cut, it will cut no the blood, the area where the blood will come out, the same area the parasite also get inside into the body of the human. The same thing will be applied to the malaria also. The same thing will be followed by the mosquito also. Clear? I think hope you're getting how the infection will be made. This is the thing have been mentioned in this slide. Now, how it becomes multiply. Now the parasite has been entered into the human body. Now all the stages of parasite in man are extracellular as they present in the blood plasma are not inside the blood cells. Okay, everything will be extracellular outside the cell, not the inside any of the blood cells, because their inhabiting area is blood plasma. Okay, in human blood, we know what is the form, metacyclic form, which is the infective stage, which is devoid of the free flagellum. It has no flagellum, become transformed into a long slender form, which is with the flagella. See, in metacyclic forms, they are devoid of a free flagellum. Now, this have been attained the some of the maturity, some of the structural changes or the developmental changes, it regains its flagella. Now, once it regains its flagella, they can move from one place to another by beating of their flagella. And the vibratile movements of the undulating membrane, we know that flagella has different types of movement mechanism, that is undulating movement, paddle stroke movement. In that undulation, because undulating movement it have been present in the trypanosoma gamete. We have studied in we have read in the structure. So you, because of this membrane, the movement can be observed. And once it reaches the area where it is perfectly okay for the reproduction, they are undergoing their asexual reproduction, that is binary fission. Now, the after it has to after it has been multiplied, it needs to develop. For that, it needs energy. For that, to obtain the energy, they are undergoing the anaerobic glycolysis. Okay. Fine. Now, metamorphosis. After it gains some of the energy, it has to undergo some of the structural changes. See, when the absorption of glucose ceases due to the antibodies, see, initially it needs energy for its growth. And after some time, when the parasite forms, the energy is sufficient, then the absorption of glucose will be ceases. How it stops? By the antibodies. I have explained already. This glycolysis, the taking of energy from the glycolysis will be stops due to the antibodies which is present in the blood. And the glycolysis is hampered and the trypanosoma stops multiplying. Once the energy supply will stop, the multiplying of trypanosomes will also stop. Now they string to short stumpy forms. They are divide of the free flagellum. Now during this transformation from long slender forms to the short stumpy forms, when the energy supply is high, they become long slender forms. When the energy supply stops, it becomes the short stumpy forms. In the long slender forms, it has a flagellum. In the short stumpy forms, they are devoid of the free flagellum. Now, the intermediate forms with somewhat shortened body and the small free flagellum also appear. The intermediate forms means the intermediate between the long slender and the short stumpy form. They also appear. Now, the intermediate forms with somewhat shortened the body and this small free flagellum also appear. These things are not that much important. You have to understand As I said in the metabolism slide, once the glucose, they are getting their energy by the glycolysis and the Krebs cycle. Glycolysis is anaerobic in nature here, but the glycolysis also takes place in aerobic form, but in trypanosome, they are following the anaerobic glycolysis. And they are also getting their energy from the Krebs cycle. Krebs cycle is happens in the mitochondria in the aerobic form in the presence of oxygen. You have to remember that after it stops the glycolysis, taking their energy from the glycolysis, due to the presence of antibodies, they are stop multiplying, and after they are undergoing some of the structural changes, that is from short to long to short, or the intermediate forms, and with the flagella, without flagella, these are all the structural changes undergoing. Now, the intermediate forms as well as the short stumpy forms depends on their energy supply on the aerobic oxidation of the pyruvic acid. As I said, when the energy supply is high, it becomes the long slender forms. When the energy supply is low, this becomes the short stumpy forms. When the energy supply is a moderate, it becomes the intermediate forms. Anyhow, they are going to convert from the long slender to the short or the intermediate based on the energy supply. Now, the stumpy forms, we know that short stumpy forms is said to be as the critidial forms. Now, these forms do not feed, ultimately die if they are not sucked up by the tsetse fly along the blood mean. Okay, it may be survive only for a particular period of time. If it's not taken by any of the blood sucking insect, it becomes dead. Now, relapse of the infection. Relapse of what is meant by relapse of infection means after some period of years, if the parasite they keep on within the body of the human or the tsetse fly, it doesn't feed any. For example, say consider this as a host organ. Now the parasite is here. It is not taken by any of the tsetse fly. Okay, it is not taken by any of the host, consider like the host. Okay, it is not taken by any of the host, it is present here, and if it is also destroying, but the spores will be, if the spores can be present, once the favorable condition will be there, they can attain their sexual maturity, and if it taken by any of the host, it can again come back to the infection. That is called as the relapse of infection. See, it has also been reported that some of the long and slender trypanosomes do not undergo transformation but change the antigens of the blood to which the host have been produced antibodies. Just they are keeping them alive until any of the vector will take. These altered slender forms survive and continue to multiply in blood, leading to future relapses of the infection. Once again, the infection can be, once it has been completely cured. See, if I have infected with this tsetse fly means I have cured, but this long slender form will continue to multiply in the blood, and they have been cause some of the infection in the future. That is called as the relapse of infection. Now, I have explained the life cycle. This picture is very important. If you know this thoroughly, you can easily know these things. Now, transfer to the tsetse fly. When the tsetse fly sucks the blood of the infected person, it also takes in the short stumpy form. See, from here, what is the form they are taking? Metacyclic form. But here, what they are taking? They are taking the trypanosomic forms. This is the things you have to remember. What is the form? They have been converted from the insect to the human, from human to the insect, and it is the stumpy forms which continue to develop in the vector. This is the trypomastigates, which is enter into the insect body of the insect, and they are going to develop the trypomastigotes into the procyclic, and procyclic to the epimastigote. And these are all happen in the gut, and from the epimastigote they have been converted into the metacyclic trypomastigotes in the salivary glands, and from the salivary glands through the bite of the insect to the human, they have been converted into the bloodstream, enter into the bloodstream. There it becomes the sexual maturity. They are undergoing the reproduction, binary fission, and the cycle continues. This is the transfer to the tsetse fly from the infected individual. Here, development in the midgut. Now, once it has been entered from the human to the insect, it reaches the midgut. Now, further development proceeds in the insect's midgut within the peritropic membrane. Okay, this is the membrane of the intestine. Now, Here the parasite transforms into a long slender forms and multiply by the longitudinal binary fission. We know that. Now, the kinetoplast moves farther from the posterior end of the body. You have to understand what is the as I said, as I mentioned earlier, what is the trypomastigotes? See, epimastigotes. Once again, I'm explaining this trypomastigotes, epimastigotes, and all. Leishmanial, that is amastigote. See, I have to focus these things. Okay, trypomastigote. See, this is the trypomastigote, where the kinetoplast is present. See here, where the flagellum is seen here, where the nucleus is here. See, every structure is present in every forms, but the location is different. Based on their location, they are named them. In trypomastigote, it is also called as the trypanosomid. Body will be elongate and slender. Here, the blepharoblast and the kinetoblast situated near the posterior end. See, this is the posterior. Always the flagella will be present at the anterior end and the nucleus will be at the middle. This is called as the trypomastigote or the trypanosomid. In the epimastigote, this is called as the critidial form. Okay, here the flagella will be present, but the flagella will be present and see the presence of the nucleus. It is at the posterior end. But this blepharoblast and the kinetoblast will be present located or near the anterior end. Just the location or the position is different, but all other structure will be present. And what about the promastigote? Here the body is elongate, the nucleus is large, and the blepharoblast and the kinetoblast will be present at the anterior end. The flagella is short and unattached to here. See, when I said the flagella is unattached and attached, see the structure here. Here the flagellum is unattached to any of the body or the pellicle. Here the flagellum is attached to the pellicle. Clear? Lalita, clear? This epimastigote is also called as the critidial form, and this promastigote is also called as the leptomonad. Okay, fine. Okay, now these are all the structural changes will be happening here, and the energy yielding process is related to the mitochondrial oxidation of the pyruvic acid. We know that mitochondrial oxidation, as I said, this is the oxidation. Definitely it is an aerobic process, but glycolysis is an anaerobic in the trypanosoma gamete. And what are all the things developed in the salivary gland means these long slender forms make their way into the salivary glands via the esophagus and the mouth parts of the insects. Via the esophagus and the mouth part of insect, this epimastigotes or the critidial form will be enter into the salivary gland. Now the critidial form will be enter into the salivary glands. There it undergoing a structural changes that is called as the metacyclic trypomastigotes. This is considered as an infective stage. Now here they metamorphose into the critidial forms with the shortened body, reduced flagellum and the kinetoplast in front of the nucleus. You have to remember the structure. Okay, once you remember the structure, you can easily know which structure will possess which type of characteristics. Now the mitochondria develops an extensive network of cristae. Now once it obtains this cristae means it becomes active. Now, the parasite respires more economically. As I said, once it regains its structure called a cristae, the mitochondria becomes active. If the mitochondria wants to become active, besides the cristae, it also needs oxygen for its survival. So it has to, at this stage, they are going to respire more as the blood glucose gradually declines. See, as I said, glucose is converted into the pyruvic acid with the process of glycolysis. Okay, this is glycolysis. And this is an aerobic process. These are all the things you have to remember. This is the aerobic process. But the pyruvic acid will be converted into the energy of the acetyl CoA in the Krebs cycle. This is the Krebs cycle, and this is happens in the mitochondria. This is aerobic respiration. Don't forget these things, write this. Aerobic respiration. See, now what is happening? The mitochondria becomes active. If the here the mitochondria loses its cristae. When glycolysis happen, the mitochondria loses its cristae. If it loses its cristae, it becomes inactive. If it is inactive, it doesn't need any of the oxygen. So this is an aerobic process. Glycolysis is an aerobic process. Now the mitochondria will be active here. Mitochondria will be active because it regains a structure called a cristae. Once it becomes active, oxygen is it keep on respire vigorously. Now it is becomes an aerobic condition. Automatically, the blood glucose level will be fall down. That is what explaining in this point. Clear? Now, the critidial forms multiply in the lumen of the salivary glands and they transform into a slender metacyclic forms. As I have mentioned here, the critidial forms, that is epimastigotes, will be converted into the metacyclic trypomastigotes. At this stage, the mitochondrial activity is again suppressed. Okay. See, in the critidial form, metacyclic mitochondria is become active. Once the critidial forms is converted into the metacyclic forms, again the mitochondria loses its cristae, it becomes inactive. When the tsetse fly bites a healthy person, it transfers the metacyclic forms. So this metacyclic forms will be entered into the humans. Obviously, this metacyclic forms is considered as an infective stage. Along with the saliva into its blood where they have been initiated another infection and the cycle will continue. Sleeping sickness. Symptoms and the pathogenesis. What are all the symptoms they are going to cause in the sleeping sickness? And the question asked here is what is the name of the disease caused by the Trypanosoma gambians? And you have to remember and the disease caused by this parasite is sleeping sickness. Once that insect will bite the human means it causes the local irritation. This is the basic symptom which subside after a few days. Only after a few days it will know. The infection is caused by the inoculation of the trypanosomes. Always there is an incubation period of any of the virus, any of the parasite. There always has been an incubation period. Only after the incubation period, the symptom will be known by the symptom will be recognized by the humans. Besides undergoing the development in the blood plasma, they also invade the lymphatic glands causing the swelling. This is the second. First symptom is irritation. And the second thing is when the immune system will act against that parasite, it causes the inflammation or the swelling. Now, the disease sleeping sickness is caused when the parasites invade the cerebrospinal fluid of the CNS. Once it reaches the spinal cord, that is CSF, cerebrospinal fluid, only then the disease sleeping sickness will caused by the parasite. An irregular recurrent fever is the first symptom of the disease. This is the first symptom. Fever. Another symptom is weakness, loss of weight, anemia, increase in pulse rate. Why it causes anemia? Can anyone say why it causes anemia? Any guesses? Just by seeing the life cycle, where the place the parasite have been inhabit in the human. As it inhabit the bloodstream, it is going to consume what? Blood. And it means it is the place where the parasite will inhabit is plasma. Obviously they are going to lose our blood, so it causes the anemia and increases in the pulse rate, severe headache. In due course the patient falls asleep first at regular intervals, then lies prostrate in death is always the ultimate fate, and this is fatal. Yes, it is living in the blood plasma, so it causes the anemia. See, death is always the ultimate fate, which means what? This parasite is fatal. Okay, and how can be diagnosed and how this parasite, this disease can be diagnosed means detected in the fresh or the stained blood films. Did you see what is the diamond medium? I have gave you the homework yesterday. What is about diamond medium? And diamond medium is the area where which protozoan can be grown. What is diamond medium and which protozoan can be grown there? And just tell me what which protozoan can be grown in the diamond medium. Yesterday I have said you, right? I have said you yesterday. Entamoeba histolytica. I have said you, right? That is the diagnostic mechanism that culture has been stained with the help of diamond medium. Okay, and that is the medium where the entamoeba, very good. The entamoeba histolytica will be grown. Like in the same way, this trypanosoma can be grown in the fresh or the stained. About the diamond medium. See, that's just like an agar medium. Agar medium is a source of many bacteria, fungi. Like in the same way, this blood film is the area or the medium where the trypanosoma can be grown. They have been taken the sample and they have been cultured in the stained blood films. Okay, and they have taken the sample and they have been cultured in the diamond medium. Only in the diamond medium the entamoeba histolytica can be grown. Okay, and based on the nutrients present in that medium they have named like the diamond medium where the entamoeba histolytica will be grown. Clear? Now, extracts of the large lymph glands, as this parasite will be in the bloodstream on the lymph gland, they are taken as their source. And in sleeping sickness stage, the examination of CSF obtained by the lumbar or the cysternal puncture becomes necessary. Okay. Now, treatment: the Gambian or the African trypanosomiasis. This you have to remember, this can be treated in its earliest stage, but once it reaches the CSF, cerebrospinal fluid, we cannot treat them. It is ultimately fatal. See, what is the thing present in our lymph glands? Why swelling lymph glands, ma'am? What is the mechanism of we human beings contains in the lymph glands? There are many secondary immune system. We have an immune system. Remember that when any parasites enter into the body of the human being, we should not think the life cycle of the parasite only. We also think what is the response that immune system of humans will respond against that parasite. The basic thing will give the basic primary immune response will be act against the parasite is inflammatory response. Okay, and what is the main thing will cause inflammation means prostaglandins, and that is cause the inflammation. And after the primary immune response, there are many secondary immune response also present in the lymph nodes is the primary secondary lymphoid organ. Okay, that is why it causes an inflammation. Okay. Once it reaches it is in at the initial stage, if any disease can be treated in the initial stage. But once it is in matured stage, and in case with respect to the trypanosoma, when it reaches the CSF, it is very hard to control. Now, Suramin, sodium, Bayer 205, etoxyl, triparsamide, they are the drugs, they have been used in their early stages to act against this parasite. Arsino, phenylbutyric acid, germanin, pentamide, this is also a drugs have been used at the initial stage to cure the diseases. Arsenine is fairly effective in cases where the CNS is involved. In the advanced stage, this drug is becomes active. Melarsen oxide is rapid in action and it is less toxic. Okay, and how we can be prevent? It depends upon the eradication of the vector. If the vector have been eradicated, what is the vector here? Let's see, fly. If it is eradicated, we can be prevent the disease. The flies can be eradicated. See where what we have studied, the flies always prefers to live near the rivers and lakes where the forest area, and by clearing out the bushes and low trees along the rivers in the endemic areas, if we eradicated the flies, the diseases also can be eradicated. And how we can be clear these bushes? By spraying out the DDT over the bush areas in the vicinity of the village. It will be effective. And domestic and wild animals, as it is act as a reservoir host, we maybe put as a whole cell slaughter, but that is impossible and it is unethical also. But this method is unethical. But if we destroy the reservoirs, there is no passage of the parasite. Okay, but that is unethical. We should not do that. Further protection is obtained by injecting 1 gram of suramin every 2 to 3 months. We have to take this type of vaccination. We can set as a vaccination. And herd immunity is the ultimate immunity. What is herd immunity? The immunity which have been see, recently we are infected with the coronavirus, right? Once the coronavirus will affect the humans, will suffer from the symptoms, of course will suffer from the symptom. After yes, of course, will attain the self immunity. Only the herd immune system will plays a major role against any of the diseases. If we once the COVID will affect the humans, of course we are going to suffer from the symptoms, and after we are recovering, when the second time the COVID will be infect, our immune system has the memory cells, and that memory cells will know how to defend this virus. That is called as the herd immunity. Okay, and the diseases also caused by the trypanosoma gambiensis, trypanosomiasis, the same thing caused the disease called the West African sleeping sickness. Also, it has been started the initiation by the bite of the tsetse fly, and the infection always started to from the when it reaches the bloodstream, and after it reaches the bloodstream it regains the flagella and they move from one place to another and they cause the lymphatic glands swelling. And this diseases sleeping sickness is caused when the parasite invade the when it reaches the CSF cerebrospinal fluid. This is fatal, and the initial symptom is fever. Only it can be treated only in the early stage, not in the later stage. That's all about the trypanosoma gambians. Any doubts? If you have any doubt means you can ask me now. Tell me fast, we have to start malaria and we have to solve some of the questions. Any doubts? Clear. Okay, fine. Then answer for this question. Which one of the following parasite is responsible for the cause of African sleeping sickness or Gambian's fever? Okay. I'll explain slowly. Of course, Trypanosoma species. See how the question have been asked. To answer this question, you have to know the name of the parasite which is going to cause these diseases. As I have said, when we read any of the diseases, the first thing you have to read is which causative agent or which pathogen is going to cause this type of disease. So name of the disease is very important also. Common name, Gambian's fever. See, we have read this as the African sleeping sickness. And if you know this is the trypanosoma, you can easily answer. But if they ask which of the following parasite is responsible to cause Gambian's fever, only if you know the species name, only then you can answer this type of question. Now, in this question, they have asked straight away African sleeping sickness. So you can easily say trypanosoma. But if you know the full name trypanosoma gambians, only then you can answer. If they ask the question, which of the following parasite is responsible for the cause of Gambian's fever? Okay, so it is very necessary to remember the name. And second question, tsetse fly is a vector for the sleeping sickness. Which of the following parasite is the infective stage? Transmits the infective stage. And what is the name of the infective stage? Of course, the answer will be Trypanosoma gambians. And what is the name of the infective stage? And that infective stage will be present in which area of the insect? We know that tsetse fly is the one going to carry the disease. And where is the, what is the location? Tell me quick. I have explained multiple times and I have stressed this point. Which is the infective stage of the disease? Which form? Metacyclic form. Okay. And that metacyclic form is the one present in the salivary glands. That is the one transferred to the bloodstream of the human, which is an infective stage. Clear? Fine. Next. Which of the following Losina palpalis is a vector for? Okay. Gambian fever. No doubt in that. Trypanosoma is Trypanosoma is non-pathogenic, monogenetic, digenetic, facultative. Digenetic. And what is the two different hosts needed to complete their life cycle? List them. Very good. Man and tsetse fly. And man is the vertebrate host, tsetse fly is the invertebrate host. And man has the which form? You have to remember which form has been seen in the man and which form is which form is present in the tsetse fly. Trypanosoform and cryptidial form. Both will be present in the tsetse fly and their location. You have to remember these. Okay, that's all about the questions regarding the trypanosoma gamins, and these are all the questions for malaria. After finishing malaria, we'll discuss these questions. Okay. Okay. And if you have any doubts, you can ask me side by side. Now we are going to start the most important topic. This is malaria. And there is a lot to discuss in this. Okay. Malaria. It is an infectious disease. We know that has been spread by the mosquitoes. Here, like the tsetse fly, mosquito is a carrier. This fly is a carrier. But what is originally the parasite? Female Anopheles mosquito. Sorry, virus. Okay. This mosquito will carrying the virus. That is the one cause the infectious disease called malaria. This is caused by the species of the Plasmodium. This is the sporozoan parasite. Once we have, when I have, yeah, after finishing the PPT, after finishing the class, I'll upload the PPT in the group. Don't worry. Okay. Transmitted through, it has been transmitted. I have explained when I taught about the classification of protozoan. In the protozoan, we have a different classes, that is Sarcomasticophora, Sporozoa, Nidophora, and the Ciliophora. In that Sporozoa, we have read this Plasmodium as an example. Okay, that is why classification is also important. Transmitted through the bite of the female Anopheles mosquito. Anopheles mosquito. See, always the carrier is important, and this name of the carrier is important. That is, this is a mosquito. This carrier is very important. Okay. Now, see, in man, the parasitic attacks liver cells and the red blood cells. This is also an important thing. In man, what is their location point? What is their affecting area? Means liver cells and the red blood cells. Now, a toxic substance is hemozoin released by the parasite causing malaria. Okay, here this plasmodium is going to releases a toxic substances, and that toxic substances is called as the hemozoin. It is the most destructive in human. So this plasmodium is a is the toxic. This plasmodium is the one going to releases the toxic substances called the hemozoin, and this hemozoin is very destructive in man. It is widespread in tropic, temperate, and subtropic region. And this malaria is characterized by what is their symptoms? Fever, exhaustion, vomiting, headaches, and it also causes coma, seizures, yellow skins, and death, even fatal. like I said, there are different types of Plasmodium, different species of Plasmodium is there, and only one Plasmodium is said to be as the fetal. There are four different types of species of Plasmodium. The most important slide you have to remember. See, there are four different types of Plasmodium. And what is that? Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, and Plasmodium falciparum. Okay, and you have to remember the different names. Tertian malaria, ovale malaria, quartan malaria, malignant tertian malaria. Okay, the other names is also an important. And what are all the drugs can be used against this malaria means quinine, atabrine, chloroquine, camoquine. Mainly, the drug will end with the quin or quinone, like that. Pamaquine, Paldron, Darabrum. Okay, and this is the systematic position of the malaria. Phylum Protozoa, we know the subphylum Sporozoa, class Celiospora, Coccidia, Eucoccidia, Hemosporidia, genus Plasmodium, and the species is vivax. There are four different species. We know that. In that, we are going to focus here is vivax. Okay, and one more thing, right? Let me explain each of these things. We know what is Sporozoa, right? Sporozoa is a subphylum where the Plasmodium have been placed here, and it has been placed in the Teleosporia. The parasitic protozoa, most of which live inside the cells of their host, that is placed under the Teleosporia. And Coccidia is a subclass, and the protozoan, which are microscopic, spore forming, as it is present in the sporozoa exclusively. Yes, it is a virus infection, but it is carried by the mosquito. See, 1 minute. What is the, see, 1 minute. See, just listen carefully. You have to know that mosquito, malaria. See, I'm going to explain everything in detail. Just remember in the life cycle. See, you don't need to confuse here. Here, I'm going to explain everything. Okay, just be patient for half an hour. Distribution of, okay, let me finish this. Coccidia is a subclass. Coccidia is a subclass. It is microscopic and it is spore forming. It is obligate parasite. And intracellular, obligate intracellular parasite means that it cannot complete their life cycle without the exploitation of suitable host. Definitely, it is going to cause some diseases to human. That these type of parasites will be placed under the Coccidia. In Eucoccidia, it is the order under the Coccidia. This is the parasite live inside the cell of the animal host. Eucoccidia is a parasite. They are going to cause the live inside. They live inside the cells of the animal host. See this suborder Hemosporina. They are the suborder of the protozoa in which the gametocytes develop independently. They are the parasite at some stages. At some stages the life cycle in the blood cells of the vertebrates. They need blood cells to complete their life cycle. And the genus Plasmodium means these are the genus of unicellular eukaryotes. We know that Protista is the unicellular eukaryotes that are obligate parasites of vertebrates and the insects. Those are the things are placed under the Plasmodium. In species is a vivax species. Now we are going to see the vivax species. Now, distribution of Plasmodium. It is widespread in tropical and temperate countries where the migratory birds or the host parasites are spread all over the world. So it is cosmopolitan in distribution. Of the human infecting the parasites, Plasmodium vivax is mostly widely distributed. We know that there are four different species, and this Plasmodium vivax is mostly widely distributed, and it prevails mainly in the temperate regions. And see, this Plasmodium falciparum is confined only to the warmer parts, so it is absent in our area, the tropical region. We are come under the tropical region, and this Plasmodium falciparum, it is very rare in our region, so we malaria can be treated in our area. This Plasmodium falciparum is our in the confined to the warmer parts. It is in the temperate region. That is British type of climate. And Plasmodium ovale through widespread, it is rarest of the four. See, you have to remember which is the rarest of the four and which is the most commonly. This is the most common, this is most rare, and this is fatal. These are the facts you have to remember. And life cycle of the Plasmodium vivax. Plasmodium vivax is the most common of the human infecting malaria. Oh, I have said you it is a virus. Oh, it is a protozoan. I'm extremely sorry. Plasmodium vivax is the most common form of the human infecting malarial fever. Okay, Plasmodium vivax is the protozoan, and that is the one going to it is the causative organism of the benign tertian or the vivax malaria, which is characterized by the 48-hour cycle between the first malarial fever and the subsequent recurrence of chills and fever. See, let me explain you once again. Plasmodium vivax is the protozoan. Okay, and this Plasmodium vivax is going to taken by the mosquito. Okay, and this mosquito is the one this is going to infect the human. This is going to cause, I have said the virus instead of protozoan. Sorry, this is the protozoan Plasmodium vivax. Okay, and this Plasmodium virus is taken by the mosquito, and this mosquito is going to harm the humans. Clear? Any doubts? Okay, it is the causative agent of the benign tertian or the vivax malaria, which is the okay, which is characterized by the 48-hour cycle. Only after this incubation period, we have no the malaria. We have no, we have experienced this chills and fevers. It is an intracellular parasite. It is present between the cells, so it is called as an intracellular parasite in man, living in the RBC and the liver cells. While the extracellular in mosquito, it is present outside the cell. Okay, see, in man it is intracellular, in mosquito it is extracellular. Okay, tertian means it is the name of the bacteria. It is sorry, once again I'm confused. Tertian, it is the name. It is based on the area. Okay, clear? No. Initially, we are going to go through the fevers when the infection has been made. See, you have to remember in this slide, it is intracellular in man and it is extracellular in mosquito. And in man, they are going to inhabit in the RBC and the liver cell. In mosquito, it is living in the alimentary canal and the salivary glands. And like the Trypanosoma gambians, this Plasmodium is also digenetic in nature. It requires two hosts to complete their life cycle. It is completed in two hosts, man and the mosquito. Okay, so let me explain you this diagram. Just remember carefully. If you know this, you can easily answer any of the question they have been asked with respect to the malaria. Okay, see, consider this area is the mosquito. This is the life cycle where the parasite is going to occur the changes in the mosquito, and here it is human. Here it is human and here it is mosquito. Now, In the salivary glands of the mosquito, the sporozoite is present. Okay, and this is the sporozoite when the mosquito will bite. As I have mentioned, the same mechanism will happen in here also. Consider this as the mosquito, and this is the hand of the human. Once it going to puncture with the help of proboscis, it makes a cut here, and through this cut, the blood will be came out. As the heparin is not present outside of the body, once it makes into contact with the air, it starts to coagulate. So to prevent this coagulation, the mosquito will send some of the saliva to it. The saliva contains anticoagulating agent which prevent the blood from the coagulation. And when it sends some of the saliva to prevent the coagulation, the saliva also contains some of the parasites, that is Plasmodium species. Any of the Plasmodium species which is present inside the saliva, when the mosquito will send some of the saliva, the parasite will come along with it. And when it come with it, through the gut, the plasmodium will enter into the body of the human. Okay, and this porocyte, it is haploid in nature, and this porocyte will undergo some of the structural changes called the merozoite. And this merozoite, takes place in the liver. See, you have to understand what is the organ which is involving in the life cycle of mosquito in the human, also in the mosquito. In mosquito, salivary glands and gut. In humans, liver and the blood are the targeted areas of the parasite. In liver, these sporocyte will converted into the merocyte, and after this merocyte will enter into the bloodstream. From in the merocyte, they are being undergoing to the tropocyte, and this tropocyte will again converted into the merocyte, and there the reproduction will be continuous. And from the merocyte, the gametocyte will be produced. Gametocyte means gametes, male and the female. Now this gametocytes will be enter into the mosquito, and here the fertilization will occur. After the fertilization, the zygote will be formed at the end product of the as the end product of the fertilization. The zygote will be formed. The zygote will be now converted into the oocyst or the ookinete. Now this from this ookinete, this have been converted to the sporocyte, and these are all happens in the gut. Now this sporocyte will enter into the salivary glands, and they have been keep on waiting for the host to end to complete their life cycle. Now another thing you have to remember is here the main host is what? Mosquito. Why? Because they are undergoing the reproduction. See, in man it is asexual cycle. In mosquito it is undergoing the sexual cycle. Clear? Do you have any doubts with respect to this flow chart? You can ask me now. Okay. Let me explain the theoretical part. Don't use any type of annotations, please. See, in asexual reproduction is passed in man in two phases. Okay, and what is a phase? First phase in the liver cells. These are all the asexual cycle. First phase in the liver and the second phase in the blood. The two in the erythrocyte. That is RBC. In the first phase in the liver cells, it is called as the liver schizogony because it takes place in liver, and it involves in the multiple fission forming the merocytes at the end. It is here the asexual reproduction is multiple fission, not the binary fission. In binary fission, only one parent cell will give rise to two daughter cells, but in multiple fission, one daughter cell will give thousands and thousands of daughter cells. That is the difference between multiple fission and the binary fission. Here it follows the multiple fission in the liver, forming the merocytes. Okay, and here in the second phase is completed in the RBC. That is why it is called as the erythrocytic schizogony, because RBC it is also called as the erythrocytes, and this phase which is takes place in the RBC is called as erythrocytic schizogony, which forms the gametocyte at the end. See, the things which you have to focus here is name of their host, mosquito and the human. And what is the target areas? Salivary glands and gut in the mosquito, and the liver and the blood in the humans. Clear? And what is the end product of each target area? Merocyte is the end product of the liver. Okay, the merocyte will be formed at the end of the liver, at the end in the liver, and this merocyte will be enter into the blood, and what will be produced in the blood as the end product of the asexual cycle, gametocyte. And after the gametocyte will be enter into the mosquito as a result of fertilization. What is their end product will be in the takes place in the gut? Sporocyte will be the end product. So this end product will be an important, you have to focus this too. In liver cysogony, merocytes will be produced at the end. In the erythrocytic cysogony, gametocytes will be produced at the end. And mosquito sexual cycle is completed in the female Anopheles mosquito. Okay, so this sexual cycle, this you have to remember. Asexual cycle will be takes place in the human. Sexual cycle will be takes place in the mosquito, and it involves two phases. Like in the asexual cycle in the human, gametogony and the sporogony. Gametogony here at the end product in the blood of the human is gametocytes. The male and female sex cells will be separately. Now this gametocytes will be enter into the mosquito. This gametocytes will be produced concerned with the protection and fusion of the gametes. What did you ask? When mosquito bite, man spores are released in the blood. How to move to liver? Kindly explain. See, just up to this, you have to know what are all the things I have mentioned. All other process will be followed, will be explained in the upcoming slides. In this slide, okay, how it has been transferred from one place to another. It loses its tail-like structure. If for the movement, it has some structure. Everything will be explained later in the upcoming slides. Okay. See, it involves two phases in the mosquito. One is gametogony and the sporogony. Gametogony is concerned with the production and the fusion of the gametes. And the sporogony is the post-zygotic multiplication, which means what? Sporogony will be happened here, where the sporocyte will be produced. After the zygote is formed, the sporogony will be occur. That is why it is called as the post-zygotic multiplication, resulting in the formation of infective individuals, the sporocytes. Here, the infective stage is sporocyte. In Trypanosoma gambians, the infective stage is metacyclic form. Here, sporocyte is an infective form. This can be asked in the question. This is the most important slide. Now, the asexual cycle of the Plasmodium vivax in the man. Now, see, in the blood meal, when the mosquito will bite, when the mosquito bite, the proboscis, the salivary glands, along with the saliva, the parasite will enter into the human. Clear? And now it reaches the hepatocyte. What is meant by hepatocyte? Means liver cells. Liver cells is called as the hepatocyte. After 30 minutes, it will reach us the liver cell. In that liver cell, they are going to convert into the zygont. That is the asexual reproduction mechanism, zygogony. It is the form of the multiple fission. I have explained into here. I have explained in the second class of protozoan reproduction. Zygogony is an asexual reproduction mechanism. Also, it is the type of the multiple fission. In this zygosome, this is the infection of the liver cells. Now, when the reproduction will be takes place, that is the asexual reproduction, zygogony takes place. Now, merocytes will be present. After the reproduction called the asexual reproduction called the zygogony, it is going to gives rise to many of the merocytes. Now, these merocytes will enter into the RBC. Now, the RBC will undergo the infection. They are going to enter into the bloodstream. There they have been converted to the tropozoite. This tropozoite, again they undergoing the process of zygogony, that is the type of multiple fission. They are going to release multiple tropozoite. Okay, now this tropozoite will convert it into the gametocyte. That is, in gametocyte we can get two types of gametocyte. microgametocyte and macrogametocyte. Microgametocyte, which is small in nature, and that small gamete is said to be as the male. Macrogametocyte is considered is the largest gametocyte, and this considered as the female. Now this again the mosquito will come into a picture. Now here the gametocyte is ready. After the mosquito will bite the infected human, the mosquito will get the infection, and this mosquito inside its gut, their fertilization will be takes place. This male and female will fuse together to form a zygote. The process is called as the fertilization. Now this zygote is converted into the ookinete, and they are going to crosses the midgut wall, and they reaches the salivary glands. There happens the sporozoite. This ookinete will be converted into the sporozoite. Uh, under the process, they are going to release many of the sporozoites. Again, in the salivary glands of mosquito, again they are going to contain the infective stage called a sporozoite. When the bite of the normal healthy person, again the mosquito will continue the infection. This is what happening in here, and the cycle will continue. Clear? This is the asexual cycle of the Plasmodium vivax in the human. See, an healthy person acquires infection when the mosquito will bite, and the mosquito contains the sporozoites. It is an infective stage present in the salivary glands of the mosquito. When the mosquito bites the normal healthy person, it transfers the parasite to the normal healthy person, and the cycle continues. Now the mosquito punctures the host skin by the proboscis. We know that it introduces some saliva into the bloodstream. Why it introduces some of the saliva into the bloodstream? Uh, to make the blood prevent from the coagulation. And when the saliva enters, along with the saliva, the parasites will also. The parasite considered here is what? Sporozoite, an infective stage. Along with the saliva, thousands of sporozoites contained therein are also being inoculated. The purpose of pouring saliva is to check the clotting of blood, as it contains an anticoagulant, okay, to prevent the infection and to prevent the coagulation. Now, sporozoite. Now see the picture. This is the sporozoites will look like. This is the pellicle. We know that pellicle gives the definite shape and structure to the protozoan pellicle. And here the convolutal tubules is present, mitochondria to serve the energy, and the micropyle. I'm going to explain what is the function of the micropyle, and what we know that mouth is also called as a micropyle. Cytophyte, and the nucleus is also present, and the paired organelles is present here. Microtubules of the pellicle. What is the function of the microtubules of the pellicle? They are going to helps there in the locomotion. There is an apical cap is present here. Now come to the theoretical part. See, the first and foremost important point when it comes to the sporocytes, it is the infective form of the parasite, and the structure is not important. And I have to ask you one question. See, if a person contains one genetic disorder, and that if the person contains that particular genetic disorder, the malaria cannot be caused, the malaria cannot be experienced by the human. What is the name of the genetic disorder? If I have that disease, means I cannot infected by the malaria. What is the name of the disease? What anemia? If I have if I have the genetic condition, means what anemia? Full name. If I have the genetic disorder, means I cannot experience malaria. The mosquito can't infect me with the malaria. What? What is the name of the genetic disorder? Anyone remember? Sickle cell anemia. See, if a person have experienced with sickle cell anemia, means RBC cells not produced. Can you bit more clear? See, if a person, what is the main area of the parasite to complete their asexual cycle in man. See, blood is the major area, that to RBC. Blood is the major place where the malaria can be converted to the gametocytes. This merocyte is converted into the gametocytes, takes place particularly in the RBC. In sickle cell anemia, in sickle cell anemia, the blood, initially the blood is in the bicoid shape. Blood is in the erythrocyte, RBC in the bicoid shape. Okay. And if the sickle cell anemia means the blood cells will looks like this. There is no place for the mosquito to convert this from merocyte to the gametocyte. In sickle cell anemia, already the blood cells looks like this. Then how can the malaria convert into the gametocytes? That cannot be possible. So the malaria, the parasite will be destroyed. So if a person experiences sickle cell anemia, he can't affected with the malaria. Okay, this can also be asked in the question. Okay, now this sporocyte, there is a spindle formed, slightly curved or a sickle shaped uninucleate organisms. Uninucleate organisms. It has one nucleus and the length is not very important. And external covering is the film elastic pellicle, which contains a longitudinally arranged contractile microtubules. Always the microtubule is associated with the motility movement, and the microtubules will be present in the pellicle, and that movement of the contraction and relaxation of that microtubules will cause the pellicle to move. Finally it results in the wriggling movement of the microtubules is the only only reason why the sporocytes can move. So the special structure, uh, so that the sporocyte can move, is contractile microtubules of the pellicle. This is why the parasite can enter from the blood to the liver to the blood. Okay. Now, this is the anterior end, there is an apical cap. This is an apical cap made up of 3 or more contractile rings into which open the secretory paired organelles. It has a uh, two or more contractile rings into which the secretory organelle. This is then organelles through from the apical cap. This organelle will be imaged. These are supposed to secrete lytic juice. What is they are going to secrete? The secretory paired organelle is going to secrete the lytic juice. This juice is going to facilitate the penetration into the liver cells. Now, can you get, you have asked me, Anjneyulu, how it has been released to the blood from the liver? Because of the secreting lytic juice and because of the movement caused by the contractile microles, everything will be possible here. Okay, now they are going to secrete some of the lytic juice to facilitate penetration into the liver cells. And now the micropyle represents the cytosome of the, okay, the micropyle represents the cytosome of the other protozoans. Here, there is many convolutal tubules of unknown function are found along the whole body of the sporocyte. If any structure is present in any of the body, definitely there has to be some function. But we should not say that this convolutal tubule has no specific function. We don't know that function, but there has to be some of the function. But this function is unknown, but it has been spread throughout the body of the sporocyte. There is a single vesicular nucleus with a centrally located nucleolus and a mitochondrion with the tubular cristae. Mitochondria, if it present with this cristae, definitely it is in the active form. So these are all the structure which is present in the sporocytes. Now, up to this, we'll stop up to this. If you have any doubt, means you have, ask me now, and the PPT will be shared immediately in the group. You can refer to that. In tomorrow's class, definitely we'll finish this malaria, and after finishing the malaria, once again, let me take the syllabus. See, We have in tomorrow we'll finish this protozoa, we have completed general characters and characteristics, locomotion, nutrition, reproduction in protozoa. And in tomorrow's class, I'm going to complete the protozoan diseases of man. Okay, after completing this pro malaria, in last 20 to 25 minutes, I'm going to conduct a small test. And I'm going to unmute everyone and going to ask question immediately, and you have to answer for that, and you have to analyze, and I'll upload the PPT. You have to revise all the entire information regarding the protozoa. Okay, clear, fine. Thank you. Be prepared for the test tomorrow. Thank you. You can leave now.